Good afternoon, everyone. Good afternoon. Compliment of the season. This is going to be our first lecture for 2026. It is going to be on STB 317, that is genetics and the environment. So we are going to start in the next few minutes. We just need to wait a bit so that others will be able to join us. Don't forget that this is going to be um, very, very important for our attendance. So please, in the next 2 3 minutes, we will join. By 6:05, we will start. Please. So before then, let's enjoy musicals. Okay. So once again, compliment of the season to every one of us. We are going to go straight to the business of the day. Just as we are approaching the examination time, we have to be much more of an exam focus. And that's the reason why today we are not going to waste our time. What we will just do is to look at the application of the Mendelian laws. In the last lecture we had, it was a kind of introductory lecture and basic understanding of what Mendelian's law. I told us as a kind of a reminder and revision that the first law of Mendel is just going to be is the same thing as when you are talking about the law of segregation or what is called the law of monohybrid inheritance. Having said that, the second one, which is second law, is dihybrid inheritance, or what we call the um, law of independent assortment. So today, just like I said, we are going to be more focused on. I'm going to be more business minded. So, and that's the reason why we are going to uh, go straight into the Mendelian laws of inheritance. Okay, I am going to start with this diagram. So, this is going to be verification of the first law. Verification of the first law. So when you look at what we have here, is that we are trying to cross a tall plant with a short plant. I told you about what tallness, what it actually stand for. I told you about dominance. I made mention of recessive. So when we talk about capital T, that means the capital T is going to be dominant over small t. So, and that's the reason why this one is going to be tall. So it is either it is going to be heterozygously tall, that is capital letter T, small letter t, or it is going to be homozygously tall, which is going to be double capital letters. So, and the recessive is going to be only going to be short, that is small letter t. So, and this P is representing the parental generation. That is, when you cross capital letter T, when you cross it with a two small letter, that is recessive. So what you are going to get at the end of the day is going to be all at the end of the fourth filial generation. I told you that what we mean by fourth filial generation, these are the set of first generation of offspring that is produced directly from the parental. Okay, this is parental. So first filial generation, all will be tall. So because it's going to be, if you do the crossing very well, it's going to be capital letter T, small letter t, capital letter T, small letter t, capital letter T, small letter t, and capital letter T, small letter t. That is going to be heterozygously tall. So that is why what you see at fourth filial generation is going to be all will be tall. All will be tall. You can make use of any other example. We can make use of any other example to illustrate this. You can say you are crossing red plants, you are crossing it with yellow. You want to cross green, which is going to be capital letter Y or capital letter R. So with this, so everything according to Mendel's law. This was exactly what Mendel observed during that time. Don't forget that I told you that Mendel he made use of garden pea plants to illustrate his this. So that means we will be crossing. So just like I said, we are crossing capital letter T, capital letter T, small letter t. You will see this in the in the comment section. So we are crossing capital letter T with small letter t. So the first filial generation is going to be what? Is going to be capital letter T, capital letter T, small letter t, capital letter T, small letter t, all will be tall. All will be tall. So you need to take note of that. That is first filial generation. So the second filial generation now, all these plants will now self-fertilize, will self-pollinate according to the Mendel's verification. So you are crossing, instead of you to cross, so you are crossing capital letter T, small letter t. So you are crossing it with another capital letter T, small letter t, so that the second filial generation is going to be capital letter T, capital letter T. This is going to be the first one. The second one is going to be capital letter T, small letter t, capital letter T, small letter t. So all these three will be what? Tall. That is why. So it's going to be 3:1, because that one is going to represent shortness. It's going to represent short. So that is why the first filial generation for Mendel's law, all will be heterozygously tall or heterozygous. You need to let that be established. Then the second filial generation is going to be 3:1. Okay, that is the first illustration. So the second illustration now, still on application of this first law, you will see here, you see gametes from parents. So this is the use of a Punnett square. So if I told you the other time that preferably we make use of what is called Punnett square. Punnett square means that okay, you make use of this rectangular representation, so that what you will have is you cross. Do you know why they put question mark here? One of the reasons why they put question mark here is that it can be capital letter Y and capital letter Y, or it can be capital letter Y, small letter y. It's still going to be the same. So you are crossing yellow pea plants, you are crossing it with a green. The green is going to be the recessive, so that by the time the gametes, I told you that the two gametes we will be looking at, and what we mean by the two gametes is that the first gamete from male is going to be the sperm. The second gamete, which is going to be from female, is going to be the egg. So, and I told you that when you talk about the law of segregation, that is, the presence of capital letter Y does not in any way affect the representation of small letter y. Each of them, each of these has equal probability, that is 50-50 or 1:1, that it is going to be represented. Okay, so this is the gamete. You can see that this one is yellow, this color is yellow, so this one is coming from this first seed, and this green is coming because it's only recessive. It's going to be small letter y, small letter y. So when you cross this one, cross it with this, it's going to be y, and it's going to appear to you as it is going to be yellow, heterozygous. Okay, so when you do that, it's going to be this. So this is an example that is trying to uh, that is verifying a different thing. What do we mean by test crop? We are going to see this when we go on. So then another thing I want you to know in this is that these are, whenever you are dealing with genetics, you should know that in garden pea plants, there are different characters or characteristics that you need to that Mendel actually paid his attention, and he was able to make use of it. The first one is that he considered the seed. The seed, in the seed, this shape of that was either round, like this, or wrinkled. If it was round, it was represented by capital letter R, uh, this, which was dominant. That's the reason why it is called dominant. That's why it is capital letter R. So the recessive, the alternative form of that gene, or what I called the allele for you the other time, that is recessive. So that is for the seed shape. The seed color could either be yellow or green. You need to know of all this. Seed color was either yellow or green. If it was not yellow, that means it's going to be green. And that's why in terms of color, Mendel favored the use of capital letter Y to represent yellowness, and small letter Y to represent what? Green. So you are not going to say it's going to be G. No, because that yellow, the green is an allele of yellow. So that means it's going to be small letter. So the same thing, you could also consider the flower color. The flower color could either be purple or could be white. So purple or white. So when you want to represent this, you can say, okay, this is going to be capital letter what? P. And the alternative, which is going to be the recessive for that, is going to be small letter P, not W. Or when you are making use of W for purple, you should know that what is going to be the recessive is going to be small letter W. So then another thing that Mendel took attention of, pay attention to, was the flower position. The flower position could either be axial, located in the axial region, or it can be what? Terminal. If it was axial, it was dominant, and if it was a terminal, it was what? Recessive. So the plant height, like the tall plant, and the short, just like the one we have just used as an example. Then apart from that, the pod shape, the shape could either be inflated or constricted. Either inflated or constricted. Inflated means it is wide. Constricted means it is narrow. That is another word. Then the pod color. The pod color could be green, whereas it could be yellow. You can now see that it's quite different from, it is the opposite of what happened in the shape of the seed. Uh, in the shape of the seed, the recessive was what? Green. Sorry, seed color. The recessive was green, whereas the dominant was what? Was yellow. However, here the dominant is green and the recessive is yellow. So I want you to actually take note of this. Then another thing is, I told you that in observing what Mendel, the first law of Mendel, I told you that it is the verification of the law of dominance. So what Mendel said was that when two contrasting allele means that when you have capital letter for one, you are going to have small letter for the other one. That's why they call it contrasting allele are present in an organism. Only one, that is the capital letter, would be dominant, and is going to be the one that is going to be physically present. That is, while the other one, which is recessive, is going to be what? Recessive, not going to manifest. It can be seen genotypically. So an example of that is what we have used the other time. That is capital letter T, which is dominant, over small letter t, which is short. So by that time, you will cross capital letter T with a small letter t, and you are going to see this. So now, how is this law of dominance, dominance, how is it going to be applied in our everyday life? Number one is that, for example, there are certain genetic diseases. We are going to see it. Genetic diseases that appear only when both parents carry the recessive. So what we are saying in essence is that there are certain genetical diseases, for example, that if it is in recessive form, the person is not going to be affected. But if it is present in what? Dominant form, or either heterozygous or homozygous, is recessive, is going to be. So the second law, which is a law of segregation. So don't be confused with what I said, first law and second law. In some textbook, what they call first law is a law of segregation, The second law is the law of independent assortment, whereas what this one is talking about, law of dominance, is the third law in some textbooks. So, but here in this lecture, the first law we are talking about is the law of dominance. That is um, for Mendel law to come to play, for Mendel law to be applicable, that means one of these, the allele or the gene must be contrasting. One will be dominant and the other one is recessive. These are the condition or conditionality that is going to preclude what is going to happen in verification of first law and third law. So that is what you should know. That is, dominance is the foundational for Mendel's law to be obeyed. Okay, so let's now see what happened in the law of segregation. So this is what is law of segregation. If you want to state it, I told you that when you are to explain the law of segregation, it's quite different from when you are to state it. So when you are to explain, the first thing is you will state it the way Mendel stated it. So, and this is it. Law of segregation states that the two copies of each genetic factor segregate during the development of gametes to ensure that each parent offspring attains one factor. Just like I told you, when you consider yourself as human, you are made up of 23 pairs of chromosome. 20 one of the pair, each of the pair is coming from both parents. That is, one is coming from your mother and another one is coming from your father as well. So that is it. So that is verification. Now look at this diagram. Look at this diagram. Don't forget that one thing you need to know is that when we are in genetics, meiosis must come into play because there can never be any crossing if there is no fertilization. That is why you need to know. No, and for some of you, you will remember and you will recall at what point will the chromosome lie on the equator. You can see that they are lying flat. So that means they are situated at the equator. So when they are situated at equator during fertilization, don't forget, during fertilization, one of these gametes, one will come from the father and another one will go to a different one, will go to the mother. So this is going to be the paternal. Is going to be the paternal. So, and what is going to happen is that half of this one will be divided into two, so that half of it will separate. This one will also separate. This one will also separate. This one will also separate. I want you to think. So that's the reason why, whenever we are representing crossing, whenever we are crossing, what we normally make use of, we make use of a double letter pair. So one letter that means one will be coming from the mother and the other one will be coming from the father. So that's the reason why it separates this same way. That is verification of law of segregation. Segregation. Another word for segregation means separation. Separation. Another, if you don't want to use separation, that means we are using segregation. So that is a verification of that law. Then another one which you already know. Okay, the third one is that this was at what? Metaphase. Metaphase 1. So you see that is when the chromosome will appear like this. This is going to be the small letter and this is going to be the capital letter. So during prophase 2, so what is going to happen is that this small letter y is this one. The other one, which is also letter y, is this one, but you have to pay attention to this color. So this is just to still explain what we mean by the law of segregation. So that is that about law of segregation. Let us now look at the statement. During gamete formation, allele of gene separates so that each gamete carry only one allele, just like I've explained to you. That is, it separates, segregates. So then what are the biological basis of this? I told you that This occurs during meiosis when homologous chromosomes separate. We have seen that. And an example of this is when you have heterozygous parents. Heterozygous parents, that is, parents with capital letter T, small letter t. Just like the first filial generation, just like I explained to you in the law of segregation. So whenever they now want to separate, they will now separate. They will separate. Capital letter will go to one place, will go to one gamete. Small letter will go to another one. And each one has the chance or the probability, equal probability of being represented. So I need to actually tell you that. Okay. Now, let me illustrate what I actually mean. That means we are going to cross, if you are crossing this verification of Mendel law, okay, verification of Mendel law. Okay. So it's going to be like that. You can make use of any other alphabet to be able to illustrate. You can make use of any other illustration or example in order to be able to verify that. Okay. Now let's now go to the law of independent assortment. This one is more complex. It is more complex because it has to do with a dihybrid inheritance. Di means two. Di, that is, you are considering two characters. I told you in the last class that considering two characters at the same time. Two characters at the same time. When you are considering the shape, you are also considering another character or characteristics. So it is either you are considering shape and the height, or you are considering so it has to be two characters. And that is, in this case now, when you look at this, it is a yellow in color versus round. Yellow in color and versus round. The way you are going to come across this, let me see if I can actually illustrate it for you using Microsoft Excel. Okay. Now, verification of Mendel's second law. So one thing you must know is that number one, it involves two different traits are involved. E.g., that is, when you are considering the shape versus the height. So you will know that that one is going to involve what? Two. And that's why they call it di. Di means two. Hybrid inheritance. Okay. Hybrid means that combination of two unrelated. That means hybrid. So dihybrid inheritance, that is it. That is, what you are trying to say is that the shape, if you are considering the shape can either be round, just like I told you, where R is going to be, where R represents round. That is for the shape, or wrinkle. Where small letter r now, small letter r will represent wrinkle, so that wrinkle. So versus the height, which can be tall, which is going to be this tall with small letter which is going to be what? Short. Okay. So what that means is that we are trying to look at capital letter R, small letter r. Then the other one, capital letter T, that is the height, capital letter T, versus what is the alphabet that we are going to use for wrinkle? That is small letter r, small letter r. We are using it with what? Capital letter T, small letter T. So that is that. So when you now cross the result of first filial generation, when you do the calculation, it's going to be, you don't forget that we said it's going to be independent assortment. Independent assortment. That means it is going to be more of a mathematical. So you will cross, you are going to pick, okay, it's going to be like this. Let me see. Let me try and work it well. Capital letter RR versus TT. We are going to make use of, let us try and increase the font of this so that you'll be able to see it well. Okay, we are trying to cross it with small letter r, small letter r, small letter T. That is what we want to cross it. This is going to be the parental generation. So that parental generation now, we'll now look at parental. So when you now interpret this one is going to be round, tall plants versus what? Wrinkle, short, plants. Okay, so that is the parental. Now, parental generation. The parental gamete now that we call parental, that is represented by P1. Represented by P1, that is parental. Okay, so it is going to be parental gamete, that is inside the gamete, parental gamete, sorry. So, and I told you these gametes include the sperm for male and the egg for female. So, when you now cross this, if you cross this now, you are crossing this with this. Don't forget, wrinkle short plant, you are crossing it, which is recessive. You are crossing it with versus, you are crossing it with round tall plants. So what you now do is that you pick this, then you pick one of these, Abby. So then you pick this. So the way it is going to be, I'm going to explain to you maybe in more practical section. But I just want you to know that that is how it is going to be now, okay? So that at the end of the day, this is the same thing. This is going to be the gamete, the two gametes, which is going to be our F1 generation. Our F1 generation. Okay? So, and you can also rewrite this as capital letter R, small letter r, capital letter T, small letter t. So, which is going to be, when you want to interpret this, it's going to be what? Round, tall plants. So, that's going to be the result of F1. You will cross it with another one, which is heterozygote, round, tall plants. Round, tall plants. So that is going to be, so that is how they came about what you see here. You can see, you can see, that is how they came about this one. You can see the F1 generation, it is capital letter, capital letter, small letter. So that during the process of segregation, now, that means you will now have our set of gametes. That is going to be the gamete for F2. Gamete that will, don't forget that for us to get F2, we have to crossbreed, we have to interbreed F1 with F1. So that is it. F2, you should know this formula. You cross F2 by getting F1 and F1 together. So in this one, we are crossing this. We are crossing this with this now for us to be able to get our F2. So, and that is going to be that we now have, when you now separate, you can pick it this way: RT, RT. This is going to be the set of gametes, RT. Then you can have R capital letter, and you can have RT, RT. So this one, okay, this one, let's use another color to itemize this. Okay, we are crossing it with this one, so that all of these we are getting it from this color. Okay, let's use another color to illustrate this, so that you'll be able to know that the gamete, this gamete is actually coming from another one. Okay, so versus sorry, we're using another. Okay. No, sorry, this one has changed. Let me go back. Let me pick another color I used before, so that okay. So this one now, it's still going to be the same. From this purple, from this purple color, it's still going to be the same set. Just repeat it, the same set. So, but it's going to be in a different color. I'm just using that color to be able to differentiate between them. So that when you now, this is now going to be the second law is going to be 16 2 raised to power 4. That is going to be 4 by 4. 4 by 4. That's 16. Okay, so that this one is going to be gamete. That is going to be the sperm slash the egg. The sign for this. So what you do is you now divide them into this. Okay. This one supposed to, okay. Let me insert one. Okay, so this is going to be the sperm. Gamete, the gamete, stroke, that's the egg. Okay, I've copied this. Copy it here. Then this one also. Paste it here also. Okay, now followed by Okay, let's use let's the edge be in purple. Women, ladies like purple color anyway, so let's use purple for them. Okay. Okay. Now, when you are doing this is going to be, you now cross this. We cross this one, multiply by this one, this with this, then this one with this one again with this. So it's going to be like we are crossing. This is 1, 2. This is 1, 1. Sorry, 2. I'm just trying to explain. That's why I'm putting it there. All these are not they are not genetically right, but just for illustration, in order to illustrate, so that you'll be able to explain it anyhow. Okay. Okay, this is going to be 1. It's going to be 2. It's going to be the 3, the third one, and this is going to be the 4th. Okay, so when you cross this, so I'm going to give you as a take home in the sense that I will just do it without coloring it. So when you get home, you go and separate it by color. So what I will do is, this is capital letter R with TT. This is going to be this one, that is 1 versus 1, 1 with 1, then 2 with 2, sorry, 2 with 1. So RT, 1 RT. Okay, 3 versus 1, that's going to be R small letter capital letter, that is that. Then RT, that is 4 with 1, 4 with 1, that is RRT. Okay, so I want you to go and separate this by the color, by their color when you get home. So this is now going to be 1 with 2. Okay, these 2 now, these 2, you cross it with 1, so it's going to be RRTT. Okay, the second one, that is 2, 2, that is RRTT. Okay, this 2, 3, that is RRTT. The next one, that is 2, 4, that is RRTT. Okay, followed by 3, 1, that is RR, okay, TT. 2, 3, 2, that is RRTT. 3, 3, that is RRTT. 3, 4, 3, 4, RRTT. Okay, now 4, 1, 4, 1, that is RRTT. Followed by what? 4, 2, that is RRTT. 4, 3, RRTT. Then 4, 4, that is going to be R. Oh, sorry, there's been a kind of mistake here. This is supposed to be small letter. So small letter is going to affect the whole of this. Small letter. Sorry, small letter. K, small letter R, R, T, T. Now, when you now have to interpret, you have to interpret to homozygous, then tau. This one is going to be interpreted as a hetero tau. This is going to be also interpreted as a hetero tau. This also is going to be hetero tau. Okay, this is going to be This is going to be capital letter half is representing the word round. So instead of you saying hetero, say round tall. Okay, this plant will be round tall. This is going to be round tall. Okay, it's also going to be what? Round tall. So the same thing here, round tall, round tall, round tall. This is how it's going to be in the exam. You have to, there's no way you can't escape Mendel's law. Round short. Okay, this is going to be short as well. Instead of it to be round short. So this is going to be round tall. This is also going to be round tall. This is going to be what? What's the opposite of being round? Wrinkle. So this is going to be wrinkle tall. Okay. This is also going to be wrinkle what? Wrinkle tall. Okay. So this one is going to be round tall. This one is going to be round short. So this is going to be wrinkled tall. So this is going to be wrinkle short. Okay, now if you count the ratio, the Mendelian ratio is always for F2 the Mendelian ratio is always what? 9, 3, 3, 1. So that means if you count all round tall, it has to be 9. If you count round wrinkled, if you count wrinkled tall, it must be 3. Then wrinkle round short must be 3. Then wrinkled and short, which is going to be this must be 1. So that is a verification of Mendel's second law. Verification of Mendel's second law, which is called the law of dihybrid inheritance. It's also known as the law of independence. Independent assortment. So where is independent assortment coming from? The independent assortment is coming from this place. This is where it is coming from. That is, the way this is going to be sorted is going to be sorted for you to get this one in the first filial generation is going to be like this. It's going to be independent of the other one. So that is the reason why it is called the law of independent assortment. So you need to actually know that because of our time. This is going to be where I'm going to stop today. But don't forget what I've asked you, and you are to submit immediately after you must have done it. Don't forget here that I explained to you that wherever you see red, wherever you see this orange color, this is going to be the sperm, representing the sperm. Where you are going to see the purple is going to represent the head. Now, the gamete of the A is sperm. All this one are representing the sperm. So, and all this one are representing the egg. Now, when you now cross them, because of our time, that is telling you that I cannot in any way be able to do. But let me just do one for you so that you'll be able to. So, one of these R is going to be pink, and one of the T is also going to be pink. So, identify I want you to identify them, the crossing, by their color. The one that will be coming from the egg, indicate it with purple. The one that will be coming from the sperm, indicate it with the orange color. So, and I want you to actually do that in the community WhatsApp group. You can do that, submit it in community WhatsApp group, or you can submit it to, okay, let me see, submit to my email. You do it in Microsoft Word and submit it in email. Okay, and this is my email, txy23n txy23n yahoo.com, or you can make use of okay, that is the one you should use so that I'll be able to monitor it. Okay, so submit there before immediately you must have finished doing it. So I want you to separate it with what? With their color. Okay, so um without wasting your time, I want to really thank you for your patience today. And don't forget that today we actually look at a verification of a first law, which is called the law of Mendel. I mean, sorry, the law of what? Dominance. First law, we illustrated that with this one. Okay, the second law, which is the law of segregation, we also indicated that with this one. Okay, whereas the second law, the third law, which is called the law of independent assortment, you must have also seen that. So, without wasting our time, I will have asked us to go to how does this the environment, how does it affect Mendel's law? Will Mendel law still hold when the environmental factor is unfavorable, or is it not going to hold when the environmental factors are still very much unfavorable? And what are these key environmental factors which you must know? So I can ask you, what are the effects? What are going to be the effects of nutrition, temperature, light, chemical and toxin, disease exposure, stress and lifestyle on Mendel's law? So, and that is what I want you to do. Okay, how so to make it the second assignment? How does the three okay? How do the three Mendel's law How are they affected? Mendel's law. How are they affected by the environmental factor stated above? Stated above. Okay, so and these are the key environmental factors. So the key environmental factors are what? So I want you to do that, which you can actually do immediately to be able to submit it